hydrophobic and much more difficult to handle through the cell. So the absorption, the metabolism, and the export is much more challenging and requires a lot of different sorts of mechanisms to do that. And the question is, can we model that? Can we model that system in a human absorptive enterocyte? In the lumen, and this can be, produced by microbes in your gut, for instance. These short-chain fatty acids can actually just diffuse straight through the absorptive enterocyte, and they enter the basal side of the cell through where the blood vessels and go directly into the bloodstream. And this is in stark contrast to the long-chain fatty acids. These, again, must be, they go into the cell, they must be esterified, triglyceride, and this is where they're in the chylomicrons, where then they are exported out the basal side in the lymphatic vessels that eventually end up in the bloodstream. And so the question is, how do we develop models that will recapitulate this sort of scenario? The predominant sort of fatty acid handling that's been appreciated forever is this one on the right-hand side here, where the chylomicrons go into lymphatic vessels. There's much less known about how short-chain fatty acids go to the bloodstream. In fact, very, very little research at all is how this actually operates and what's importance it is. So back to the models. How do we model this in a very physiologically accurate way? Well, historically, we've been relegated to these sorts of models. Mouse models, mice are not humans. We're learning more and more, especially as single-cell sequencing data are coming out, that there's very big differences in mice and humans. The colon cancer cells, if you're interested in studying absorption of fats, that typically doesn't happen in the colon. So you can imagine the problems associated with cells that don't even make the same types of genes, nor the same magnitude of genes. Plus, they're cancer cells, they're not fully differentiated. And then now we have 3D organoids that are generated from stem cells. So normal stem cells that will generate a three-dimensional hollow ball. And the problem with these, even though they're very powerful, is the internal apical side is inaccessible from the outside. So if you wanted to add some sort of luminal contents, some fatty acid to study how it's being handled by the cells, the only way you can get it in there is by microinjection. There are some techniques that you can flip the inside and outside of these organoids so that the apical side here with the microvilli are on the outside, but then you have the same problem. You can't access the other side. So these are some of the challenges that really led us to think about how to redesign these ex vivo models to allow us to address these really challenging questions with fatty acid handling. I'm going to take you through a bunch of the literature very quickly here that we've published in the last, I would say, probably 8 to 9 years with one of my collaborators, Nancy Albritton, who is a biomedical engineer. She was the chair of the department at UNC, and now she's at University of Washington in Seattle. But again, on the right-hand side, this is what we create, is this epithelial monolayer that contained compartmentalized tissue, meaning that you had a proliferative zone with stem cells that was separated from a differentiated zone, and two, that we could actually create all of the different post-mitotic lineages that you can find in this tissue. Now, that makes it a much more physiologic from an architecture perspective, but also probably a functional perspective and how that the contents such as fatty acids are interacting with different cell types because of that architecture and the compartmentalization of a stem cell zone and a differentiated cell zone. So I'm going to take you through how we started this. Initially, we just basically had to isolate the stem cell compartment, which is the crypt. Here's an example from mice where you can do a very simple EDTA prep. It leaves the crypts that are still in submucosa. You do a very hard shake. It's very simple, actually, and you get out this very clean, set of crypts that you can then take and try to culture. Now, the Hans Klaver's group, probably about, 12, 13, 14 years ago, developed the ability to take those crypts and to generate what we call an organoid. And this really, this technology really was, underpinned by tricking the stem cells into thinking that they were back in the body by recreating the niche where the stem cell lived. And this is done through several factors, EGFs, R-spondins, basically high Wnt signaling. But also what's required is an extracellular matrix to make contacts with the cells. If you do not have those, you cannot have a maintenance of a stem cell compartment, and this organoid will not form. So these are stem cell driven structures. They're spherical. The inside of this is dark because the cells are basically sloughed off into the center, very similar to the lumen of the gut. We call it the pseudolumen. If you made a cross section of this, you could see that these cells, these protrusions are a monolayer, and the stem cells are actually in the tips of the buds of each one of these. So they really are a tiny little mini gut. What they do not have is they do not have villi. The differentiated cells are kind of in between these buds where the stem cells are, and there's not a lot of the differentiated cells. It's mostly stem cells that keep recreating these small little balls. And since they are stem cell driven, you can dissociate those, you can make more of them. It's a very powerful system. But as you can tell, they're not very ordered. They're not very consistently ordered. And that was one of the things that we then tried to say, how can we order the cells in a better way? Two, how can we make these an easy thing for investigators to use? Because as I showed you previously, these are 3 dimensions. And the ability to image these, to follow them, to track them, to quantify them is extremely challenging and typically very low throughput, very easy to do. So we said, hey, what if we take the 3-dimensionality out of this and flatten these out? And that's what we did here. It's a little hard to see because of the lights in this. You can probably see it better on the computer screen. But we basically generated a 2D monolayer of these cells on an extracellular matrix that was similar to the 3-dimensional extracellular matrix, but we put it on a substrate that allowed it to grow flat. The blue in this case are nuclei, the green are S-phase cells or dividing cells, and the red are the Panna cells, and these are these niche-forming cells. If you look at the right-hand panels here, you can see that you've got these little Panna cells that are next to these green cells, kind of suggesting that there's a little niche formed here. But again, if you look closely, you'll notice in the larger picture that nothing is ordered. You still have this disordered array of cells. We don't have compartmentalization of stem cell zones or proliferative zones and differentiated zones in a manner that mimics what I just showed you in the schematic on the left. And so in collaboration with Nancy Albritton's group, one of her expertise is microfabrication. And they can microfabricate pretty much anything you want. And what we said is, hey, can we recreate this kind of cryptvilla structure using microfabricated collagen scaffolds? And that's what you're looking at here. All of the dimensions are the same dimensions as what you would find in vivo for human and or mouse cryptvillous axis. And these were placed in an arrayed format. You can see in the center screen here, where you have the villi tips. You're looking straight down here. Those are the large circles, and the tiny little circles in between are the crypts. There are 5 crypts that feed a villus, just like you would find in vivo. And if you look on the right-hand side, this is a side view on the bottom, and you can see the arrayed form of villi structures on the top and the crypts on the bottom. Now, this is a collagen scaffold. We have to put cells on top of it. And we would put stem cells on top of the scaffold. They would cover the scaffold entirely. And you can see in the crypt zone, this is looking straight down. You get a nice coverage of cells that are surrounding the crypts. And this is the top of the villi. And you can see a nice coverage, complete confluent coverage of cells there. If you take a 3-dimensional image of that, and in this case, blue is nuclei. In green, we've stained this structure with EDU. You can see that we have both a compartmentalized stem cell zone more towards the bottom and differentiated at the top. Now, there are proliferative cells here up the kind of crypt axis, villus axis, and you would never see that in vivo. And so we've done optimization to actually tailor the factors so that they don't diffuse as far, and we can keep the stem cells just in the crypts. And I think that's what you can see in this next picture. So this is a cross section of what I just showed you, where we have the crypt villa structure, but now we've tailored those factors, stem cell factors, so that they're only diffusing to the bottom cells. EDU again is the proliferative stem cells. You see that at the bottom here. One of the questions we asked was, does it have the same migration properties that you have in vivo? So if you look at the left-hand schematic, the stem cells are born, they give rise to a transit amplifying zone in the yellow, and then they migrate up towards the tip of the villus, and that takes only about a week. All the cells in your gut turn over in about a week's time. So if you go back to the data on the right-hand side, you'll see we pulsed on the left on A with EDU and then removed the label. And you can see that after 96 hours that those labeled cells have moved up that villus axis. So not only is the tissue compartmentalized, it also has a migration pattern and properties similar to what you find in vivo. And this quantification C of the migration is on the right. Okay, so now you have this three-dimensional system. But as all of you know, imaging something that's over a few microns in any manner is very challenging, even if you use multiphoton. And so we thought, well, this is not really, it's a great system. It's very physiologically relevant from a microarchitecture standpoint, but it's not very practical for use in labs. And so we thought, what if we take that basically the architecture and flatten the 3D down to a 2D system? And I'll explain a little bit more what that means. If we can compartmentalize, meaning separate or zonate the differentiated zones from the stem cell zones, but still keep that arrayed format on 2D, that would actually present a very nice model where you still could look at your stem cell compartments and differentiated compartments in pretty much any standard microscopy modality. So what Nancy Albritton's group did is they developed this insert. If you look at the image on the top here, and it sits inside of a transwell, and the gray is an impermeable membrane. The holes are permeable. And this sits in on the bottom of a standard transwell. We coat that with collagen. Now, if you look at the bottom, the gray area, we coat that with collagen, and that collagen is permeable just over the holes. It keeps the cells that grow on top of from going down in the holes. And you can grow confluent monolayer cells across this. So the concept is, if you can grow stem cells across this and keep the stem cell factors only on the bottom, but remove them from the top, only the cells over these the permeable holes will have access to the factors. And so this is how it works in real life. On the top is the schematic. We have WRN is the stem cell media on the top and the bottom, apical basal compartments, telling the cells just stay as stem cells, grow as stem cells, and they cover the confluence. You can see underneath it at day 2. And then after a certain period of time when they're confluent, we switch the media where we take off the stem cell factors from the top and only leave them on the bottom. You can see on the top right-hand schematic there. And now what it does is it compartmentalizes that tissue. What you're looking at here in green is a stem cell marker called OLFM4, and we CRISPR knocked that into the endogenous locus. And now we have a stem cell marker that we can trace. So on the left-hand side, you can see confluence of stem cells. When you compartmentalize the tissue, the stem cells only stay over these holes. And we can grow these indefinitely. You can grow them. I think we've grown them for several months now because there's a stem cell zone. They grow very nicely. And this is what it looks like when you look at it with some stain markers. You have your stem cell zones in the green. The magenta are just a differentiated, it's KRT20, which is a general differentiation markers. And of course, when we go and stain these for the other lineages like goblet cells, endocrine cells, best 4 cells, we can find these in these intervening regions. And like the tips of your villus, where these kind of like units come together between the stem cell zones, we have apoptosis that happens, and you can see sloughing of the cells. So it's really a dynamic living tissue that functions very similar to what we find in vivo. So that's kind of where we're at right now with the different sorts of models. And that probably, again, was about an 8-year journey to get to those models. And we're using quite a few of we call them crypt planar microarrays. Many of our studies, in fact, most of them now use that because it's such a powerful system. Now that we have these tissue constructs, what I'm going to do is show you really two things that we've used these for in fatty acid handling and also C. difficile infections to understand certain properties of either fatty acid handling or toxin, the effect of toxins on the epithelium. So how can we leverage these stem cell differentiation, human ISC, that's human stem cell differentiation, to study lipid handling? Well, again, we decided to try to make this as physiologically relevant as possible. And we went to human epithelium to get stem cells. And we get very unique research. We get the entire human gut. from transplant organ donors. So these people are as normal as possible. And we have inclusion and exclusion criteria. So we do not take any organs that have undergone any sort of trauma. They have to be serology negative for all communicable diseases. They can't have inflammation because we want as normal as possible. We can always ask for the diseased or injured tissue, but we want normal. And we get the whole entire gut. So we get, small intestine and colon. And conceptually, what we do is we isolate the stem cells, very similar to what I showed you with the mice in the last set of slides. And we plate these on a thick collagen scaffold. This is important. The thickness of this is important because the stem cell is like a very soft substrate. If you coat the plastic, for instance, with collagen and put the stem cells on, they'll undergo a few rounds of division, and then they'll differentiate. So it's got to be soft, and we expand the stem cells to pretty much an indefinite level. I mean, we can grow, plates and plates of these indefinitely without running out. And then we place, once the stem cells have expanded, we take them off and we put them onto kind of conventional transwell culture systems that have been treated with different extracellular matrices here. But importantly, for those of you who don't know how these work, these membranes on the bottom of the insert is permeable. So you have access to the factors on the top and the bottom that the cells make, or you can apply different factors to the top and the bottom, or the luminal and basal surfaces of these. And in this case, we use a fluorescently labeled fatty acid, at least a host of different ones from some short, medium, and long chain fatty acids. You can add that to the apical compartment, luminal compartment. And of course, those fluorescent fatty acids will be absorbed by the epithelial monolayer. And then in some cases, they will be exported out the basal side, which you can sample then and do all kinds of downstream assays. So it's a very nice system. And I told you before that we get these organs from transplant organ donors, and we isolate stem cells from 6 different regions: 3 from the small intestine, the duodenum, jejunum, and ileum, and 3 from the colon, the ascending, transverse, and descending colon. We have about 18 normal, healthy human donors banked right now. And we isolate and just store the stem cells because those are the ones that we can then take out and expand indefinitely. And the nice part of this is, depending on what part of the intestine, proximal or distal axis, that is doing some job, we can pick that out. So if we're interested in how fatty acids are being handled, we're going to pick something in this kind of more proximal region to put the cells on our platforms. If we're interested in C. difficile infections that happen more in the colon, we would pick colonic stem cells and generate our tissue constructs from that. So these organ donors have been very important for really developing very high resolution transcriptomic models at a single cell level that have informed how we build these models and how we know whether or not they're physiologically relevant or not. So that's a big question when you're building an ex vivo model is, yeah, it looks like an intestine, you're using intestinal cells, but is it really behaving like an intestine in vivo? Well, in order to answer that question, you have to have some sort of benchmark. You have to say, well, this is what a human in vivo looks like. How close is my in vitro model getting to that? And this study that we published, 2022, this was really our benchmark for answering that sort of question. And what we did is we took intestinal epithelium from 3 different donors and isolated into these 6 regions. For the small intestine, we mixed the villi and the crypts at a 50-50 ratio. And the only reason we did that is the villi, there's so many more cells in the villi than the crypts, we would have skewed the ratios and probably got very few crypt cells. So we kind of arbitrarily did a 50-50 mix of this, and the colon doesn't have villi, so we just use 100% of these crypts. And then we performed single cell RNA sequencing. We sequenced over 13,000 cells across all 3 of these donors. We identified 25 different lineages between the small intestine and colon. And the importance of that is now we have not only just the lineages, but we have all of the genes that mark that cell type. So we have a signature, and then we have all of the other genes that may be indicating certain mechanistic functions of those cells. So this is now our benchmark to be able to say, once we take our cells and put them onto a kind of synthetic system, how can we achieve the signatures that we're finding from these in vivo studies? Well, one of the things, since we're interested in fatty acid handling, is how do the absorptive enterocytes function? What are their transcriptomic signatures that we need to mimic? And one of the things that had been concepts that had been coming out in the literature at the time is that all of the absorptive enterocytes, so these are the main cell type of the gut that are doing the job of absorbing nutrients, that they're not all the same. They're not all created equal, that there's actually a maturation of these cells from the stem cell. If you look on the left-hand side at the bottom, moving up the villus axis to the top where it's purple, that they undergo a maturation. And it stands for early absorptive enterocyte, I is intermediate absorptive enterocyte, and M is mature. And these designations of this kind of axis of maturation was defined by trajectory analysis. And this is done with a single cell sequencing data. I'm not going to go into the details, but it's basically a computational way that looks at the number of spliced and unspliced transcripts to give you sort of an age of that cell. And if you look at the arrows, the directionality of the arrows from the stem cell, which is kind of the yellowish knob at the bottom, you can see that there's a progression towards the top, or the villus tip, which would be the mature absorptive enterocytes. And when we looked at the genes that marked certain fatty acid handling functions, so fatty acid transport, triglyceride synthesis, chylomicron formation, lipid droplet formation, fatty acid oxidation, you could see that the number of genes and the magnitude of those genes increased from left to right. So from the stem cells to the mature absorptive enterocytes. Now this gave us a set of genes and also a maturation state in vivo that we would need to kind of mimic. So we have the set of genes, and we know that we have a certain kind of maturation state that we have to achieve. So how do we do that in vitro? And that was the next question, is how do we tailor the conditions to best model these in vivo gene expression patterns for fatty acid handling? Again, we turn to our stem cells that we get from these transplant organ donors. We plated them into the same transwell system, except this time we scaled this to a 96 well system. So you can imagine the power now you have to do anything in high throughput. And then we had to ask the question, when we take the stem cells and we put them on this transwell system that we've kind of, it's a synthetic system, how close are they to what we would find in vivo? Well, the first thing we did is we asked, well, how close are our stem cells? And we grow them in the stem cell media. So if you look at the top schematic, the green cells, we did single cell sequencing on those. And then once we differentiated them, meaning we took off the stem cell media, put on a differentiation media to push them towards the absorptive lineage, we did single cell sequencing on that. If you look on the right-hand side, these are the data for that. Green are cells that were in the stem cell expansion media only, so they should be mostly stem progenitor cells, and the orange is the differentiation media. If you follow the arrows down, you'll see that the blue cells are cells, we basically had a score that was based, a stem cell score and a differentiated absorptive enterocyte score, which is based on many genes, a whole set of genes. I think it was like 20 different genes. And if you have a higher score for all of those genes and the magnitude of the expression of those genes, it's bluer. So you can see in the expansion media that most of the cells are very highly blue. And then the differentiation media for absorptive enterocytes on the right, that only the top cells that were basically the orange cluster were highly enriched for these differentiated cell genes. So this really told us that, at least from a transcriptomic level, that we were culturing cells with stem cells, and our differentiation media was pushing them towards some sort of state that was like an absorptive enterocyte. One of the functional properties of absorptive enterocytes, or basically your gut lining in general, is that it has a tight barrier function. And so we performed transepithelial electrical resistance on these cultures over time. So this would represent sort of a pseudo measure of maturation. And if you look at the bottom left-hand graph, anything in green represents the cells on the transwells that were in the stem cell media. And you can see a very low tier. So stem cells do not have a lot of barrier function. But as soon as we switch to the differentiation media, which are all of the data points in the kind of orangish color, you can see that there's a nice increase in this transepithelial resistance about day 7, but then it decreases after that down to day like 11 or 12. Now, people, when I show them this, they go, well, why are they dying? Well, you want them to die. And the reason you want them to die is these are undergoing an exact same lifespan as what you find in vivo. A 7-day lifespan is what epithelial cell will live in your gut. So this also suggests that these, whatever the mechanisms are that are defining the age of these cells are still intact in our in vitro system. And it also gives you a window where you know you actually have to do your experiments before they start to die and slough off. Probably between days, let's say 5 and 7. It gives you a 2 to 3 day window to do experiments in these. Now, each of those red arrows you see there, we took RNA from each of those time points into transcriptomics to now try to get an idea of what is the state of the cell at each of those maturation states. Remember, our goal here is to say, how close can we get to the in vivo maturation of those mature absorptive enterocytes to get a very close representation of fatty acid handling that we would find in vivo. On the left-hand side here are those transcriptomic data. Each dot is a different technical replicate. Day 0 is our stem cells. Day 2 is when we differentiated them. And you can see on the first principal component on the x-axis, there's a big jump between the day 0 and day 2 is what you would expect, because you're saying, okay, differentiate stem cell. Then for each of the days, day 2, 5, 7, 11, and 10, there is much smaller difference on the principal component 1, but more on the principal component 2. And that suggests that there's now more subtle changes that are happening after each one of these days, because the cells have now gone to an absorptive enterocyte fate, but they're now, we think, based on this maturing. And I'll show you how we kind of showed that in a minute. One thing we decided to come up with, which is, this is one of my favorite experiments that we did, is how do we know what day of maturation is the closest to what we find in vivo from a transcriptomic state? So looking at all the genes that are expressed in vivo for absorptive enterocytes at those early, intermediate, and mature states, how do you compare that to your in vitro? They may not be the same, right? The number of days may not be the same, and we might not even know. So what we did is we took all of the genes that were involved in lipid handling functions, and we did this thing called the residual sum of squares. And this is just basically a statistical way of saying how similar are the genes and the magnitude of expression of those genes at any particular place where they overlap between the in vivo or the in vitro. And so you can use this as a little scale. And if you said, hey, I want to make a mature absorptive enterocyte, you'd follow on the bottom x-axis to the right, and you would follow, then go up the y-axis and find the lowest number that would represent the most similarity. That's how this particular math problem, and you would say, oh, that's at day 7. So, or sorry, day 6. So you could actually go all the way up into day 6, 7, 10, 11, and you would start getting the most similarity of your in vitro culture to those genes that you find in vivo. And conversely, if you wanted to do more early absorptive enterocytes, you would pick something around the day 0, which would be more like a stem and an early absorptive enterocyte. So now this gives us some sort of quantitative metric that will allow us to figure out how long to mature our cells on the transwells to achieve a transcriptomic state that's very similar to the state in vivo. Now, this is just for lipid handling genes. You could do this for any sort of biological process and make this kind of square and do the same thing for your predictions. On the left-hand side is really just showing the genes, how they match up. And of course, not everything is exactly the same, but the trend you'll see if you look at the left-hand kind of column, the stem cells are on the left, you'll see that there's very low expression of these fatty acid handling genes. And as you move to the right, or mature those cells, as they're getting darker. Same thing on your days in vitro on the right-hand panel. Day 0 are stem cells, and you can see as you move to the right, the magnitude of expression is going up for all of those. So it's very similar. So now that we have this kind of idea of how to get the cells on our in vitro system to kind of match the in vivo maturation state, we wanted to kind of validate, is it working like we think it is? And one of the easiest things we thought to do was to look at fatty acid oxidation. And one of the things that is very well described is the long chain fatty acids, when they get in the cell, they have to be chaperoned into the mitochondria with a protein called CPT1 to undergo beta oxidation. And so it's a very easy thing to kind of like look for if you can inhibit CPT1. Well, we have to say one is CPT1 expressed in our cells at the right day. Here again on the left-hand side is the in vivo. Blue is higher expression. You can see that even in the early absorptive enterocytes, you have CPT1A. Stem cells have very little. And then as you move to the right, the magnitude increases. We saw the same thing in our in vitro expression pattern for CPTA1. And you can see at day 7, we have a very similar sort of magnitude of expression that we have in our mature absorptive enterocytes. So this informed us that we needed to start our experiments probably on day 7 to get the closest expression pattern to that in vivo. Now, I told you that if we can inhibit the CPT1, and we can using a drug called etomoxer, what should happen is the long chain fatty acids will get into the cell, but they can't actually undergo beta oxidation to shorter chain fatty acids that then would be exported out the basal side. And that was the prediction. Shorter chain fatty acids, of course, they don't need to undergo beta oxidation to get out the basal side. They can actually just go directly through because of their increased water solubility. So these are like C8 and below chain fatty acids. So that's what we did. We put, BODIP C5, which basically should go straight through. It shouldn't be impacted by CPT1, and that's what we saw. So whether or not we had etomoxer or not, there was no difference in the export of that short chain fatty acid. That's exactly what we would have predicted because of its chain size and its solubility. And then we tried this with a long chain fatty acid. So we put BODIP C16, so it's a 16 carbon chain fatty acid, and of course read the basal contents on a plate reader. And in this case, when you inhibit etomoxer, this is the purple line, you can see that there is a definite decrease in the amount of fatty acids that are exported. So again, this is showing our system has CPT1, it's inhibitable, also producing a metabolic sort of profile of the beta oxidation that we would have predicted. But that doesn't tell you, that's just anything that comes out the bottom. It doesn't tell you the chain link. And you notice that there are some fatty acids that are coming through. And so we used thin layer chromatography to now get a signature of those different fatty acids that were on the basal side. And if you look at the left-hand side panel and the middle, these are just basically a medium and long chain. I'm just showing you 2 different carbon chains to make this point. If you look at the vehicle alone, you can see that the predominant species that comes out in both the medium chain or the long chain is C6. So there was some beta oxidation that was happening, but only C6 was coming through. And you can see this quantified in the bar graphs on the left. If you looked at the shorter chain, the C12, you can actually see some larger chain. C12 can actually be exported a little bit. It's more hydrophilic than C16, and it can actually get through a little bit. But the predominant species is C16. When you add etomoxer, you decrease this amount of C6 that's going through. So you're inhibiting that long chain fatty acids getting beta oxidized down to C6. So etomoxer is actually doing what we think it's supposed to do in this case. What's happening to these long chain fatty acids? They're not being beta oxidized, meaning the chain length is not getting shorter and shorter through the mitochondria because they can't get into the mitochondria. And so what we did is we said, well, they must be stuck in the cell. And that's what you're looking at here on the right-hand side. So the top is the vehicle. You can see that there's very little green, or BODIP C16 in this case. But in the etomoxer treated, where the fatty acids got in but could not be metabolized, they got stuck. And the resolution isn't good here, but these are basically like lipid droplets. You can look really closely, and they're making lipid droplets because they're not able to be metabolized and shunted out into either fatty acids or chylomicrons. And this is just a quantification of the BODIP C16 in these images here. So we look to say also, if we inhibit fatty acid oxidation, we should see the opposite. I'm sorry, if we potentiate fatty acid oxidation, we should see the opposite. We should see an increase in smaller chain fatty acids that are exported out the basal side. So we put B16, so the long chain fatty acid, into the system, and we added two drugs known to potentiate or increase fatty acid oxidation in other systems. And it wasn't in the enterocytes at all, but in the muscle, basically. So C75 was known to increase oxidation in the muscle, as well as metformin, which we didn't know that. It was kind of a surprise, but it happens in skeletal muscle. And so when we potentiate fatty acid oxidation, what we see is an increase in smaller chain fatty acid with both of these drugs. Black line is control with a vehicle alone, and then with both drugs, we see an increase in short chain fatty acids that are coming out the basal side. And so our system seems to be working quite well. You can imagine now, since we have this in a small form factor for high throughput, that you could actually screen drugs that will impact this sort of fatty acid handling and allow you to do some very detailed resolution studies. This, again, is looking at the basal compartment after you potentiate with one of these drugs. You can see that there's definitely an increase in the short chain C6 fatty acid that's coming out here, and that is the quantification on the right. So it's a very nice, simple way of being able to take these monolayers from stem cells, differentiate them with medias that have specific formulations, make them into absorptive enterocytes, mature them on the platform to something that's very similar in transcriptomic state to in vivo absorptive enterocyte, and then do experiments. We think it's a very highly physiologically relevant way. And one thing, my student at the time, Ismael Gomez, he did this work. He was one of these people that could like find all these papers and make really interesting links between them. And he found this paper, actually it's older than me, 1967. It says studies on the absorptive defect for triglyceride and A-beta lipoproteinemia. And in the paper, it says a considerable percentage of dietary fat is still assimilated in these patients. The route by which it is absorbed remains to be clarified. And so he postulated that perhaps what's happening in these people, they have a mutation in MTTP, which is basically doesn't allow the chylomicrons to be made. So they can't get the fat into the body, but they do have fat that's assimilated. His hypothesis was basically there was an increase in beta oxidation that was happening in free fatty acids, where short ones were able to get through, and then, of course, distribute through the body. And that might be a potential way that these people actually can distribute fats. So the take-home messages from this and some thoughts are that these human stem cells can be efficiently differentiated into absorptive enterocytes with a transcriptomic signature that resembles that from in vivo. Export of long-chain fatty acid metabolites can be manipulated by perturbing fatty acid oxidation. So this is, again, a drug targetable thing if that is of interest. Increased short-chain fatty acid export through enhanced beta oxidation may explain the escape of the effects from these MTT mutations that people with A-beta lipoproteinemia might have. Still an active area of research. And this work was published in a little more than a year ago now. And you find there's a lot more details that I have time to get into. Okay, the last bit what I'm going to talk about is how we're using these human stem cell derived cultures to study the impact of bacterial toxins. And in particular, we're looking at toxins that are made from Clostridioides difficile bacteria. These bacteria are spore forming, and they make two main types of toxins, toxin A and B. C. diff is the leading cause of gastrointestinal associated death in the U.S. Most of this happens in healthcare settings, either in hospitals or in certain types of healthcare settings for older individuals. And they're associated with certain antibiotic use. So people that get large amounts of certain types of antibiotics can acquire these spores, and they will colonize the gut, and it causes basically death of the gut in many cases. There's 500,000 cases of this each year in the United States. It's a life-threatening disease many times and causes toxic megacolon and bowel perforation. And there's really no really good treatments. The only good treatment is resection of the diseased tissue or fecal biota transplants, and these don't always work for everybody as well. So, and they're still kind of experimental in some respects. And so we wanted to actually have a model that we could start testing how that these toxins work from a biological perspective on normal human enterocytes, so not cancer cells, and try to develop a good model for drug screening to try to identify new drug modalities to treat this disorder. The mechanism of how this toxin work is basically toxin A will be internalized through a receptor interaction, and then it gets into an acidic vesicle where the membrane, it's inserted into the membrane, and then there's a cleavage of the GDT domain, and this goes up and it will actually cause glucosylation of Rho, and that will cause basically the cytoskeleton of the cell to change. When that happens, the cell shrinks, changes formation, and it gets leaky. And if you have a leaky barrier, then there's a million other things that happen that are bad for you. You get luminal contents that get into the submucosa. You have an inflammatory response. It's quite bad. And that's where these cytopathic and cytotoxic effects happen from this particular type of reaction. So we thought, hey, do our cells on this platform have the receptors for C. diff? And so we used our single cell sequencing data set, and we looked at both intestinal stem cells and mature absorptive cells. These are on the y-axis here. You can see from the three regions of the small intestine and 3 regions of the colon, both in the stem cells and the mature absorptive cells on the right. And you can see that the receptors are largely there, but at different levels. So for those of you who are used to looking at these graphs, the size of the circle is the percent of the cells expressing, and the color, the increased color, is the magnitude of that expression. And you can see that most of these receptors are expressed to some level. The frizzled receptors are much lower, but they still are expressed. And what we decided to do, because of the clinical implications of where this disease happens, and we use the descending colon stem cells to create this model. We did this the same way as I told you before. We took crypts from the colon this time. We expanded these on our thick hydrogels. We plated them onto the transwells as stem cells, then we differentiated them for 4 days. And in this case, 4 days for the colon created that tier profile that we looked at before, which was a kind of proxy for this maturation state that we wanted to achieve. We then wanted to look at the maturation here. This is colon stem cell signature from the in vivo, and you can see in EM, so these are stem cells, and then DM is differentiation media. You can see that stem cell signature is decreased, and that's what you would really predict. So conversely, the genes here are late absorptive colonic in vivo signature. So this is a panel of genes we use for that signature. And in this case, you can see that the stem cells, start out low and then they go high. And it's exactly what we would predict. We also wanted to compare this to CACO2, which is the conventional model for doing C. difficile sorts of infections, Vero cells and CACO2s, which are cancer cells. And you can see that in this, they stay way more stem-like after 22 days of differentiation here on the left. And on the right-hand side, you can see that they stay very primitive and they don't express many of the genes, or at least to the magnitude of the cells in our culture. So the CACO2 cells, despite being able to be differentiated to some extent, they still do not achieve this level that you can find in our primary cell cultures. So that's what I just showed you. So again, the next thing to do is to look at the barrier function of these cells. Again, same thing we did with the colons, we did with the small intestine. These days on the left are in the expansion media. They're basically stem cells. When we switch, we get an increase in the tier. We looked at 3 different donors. So this is a big question we get too, is are all donors created the same? And in general, I would say to some extent, right? So we do have in tier upon changing media to differentiation, but the magnitude of that barrier function is different between different people. I think this highlights the point that when you're using primary cultures that are not transformed, they're not clonal, they're very non-homogeneous populations, you really do need to look at a lot of different donors to get probably an accurate result. And again, compared to CACO2, you can see that there's really no increase in that barrier function. So if you do apply something to the system that causes a drop in barrier function, but it's already low, the question is it really that relevant? And so that's another thing about CACO2s that are challenging to use. I just have a few more slides. And what I'm going to show you is that we apply the C. diff toxin A to the apical side. And what you can see is that we have apical and basal side. You can see that we have a drop in transepithelial electric resistance, meaning we have holes that are opening up because of the toxin, which what we predict. And it's also dose dependent. So the oranger line is 10x concentration of TCDA compared to the 1x. And incidentally, the 30 picomolar is the same concentration that you can find from fecal samples from patients. So this is a real physiologically relevant dose of toxin. One thing we wanted to do to make this more high throughput or an easy way, measuring tears is it's hard. You can takes a long time. You have to have the cells that are incubated for a long time. We thought we might be able to use a proxy for the tear by looking at tight junction proteins, because these are what are disrupted when the toxin's doing its job. We made a transgenic line in our stem cells that was a ZO1 EGFP reporter gene. You can see on the top left-hand panel here that there's a nice indication of the ZO1 in between the cells. This is just looking at the endogenous protein and the magenta and the overlay. So we knew we were making a protein and it was going to the right location. And then we added the C. diff toxin A over time. And you can see that 4 hours and 8 hours, there's definitely a decrease in the signal, but also importantly, the distribution of those tight junction proteins. And so this is a faster way than doing TEER, and correlated. We have a correlation plot showing this. I didn't put it on here, showing that there's a very strong correlation of this redistribution to the amount of time that the cells are exposed to the toxin. And again, using a high throughput modality, you could put these fluorescent reporter gene cells on and screen compounds very, very rapidly. One thing we also wanted to do, and this was kind of, we didn't really expect this result, but we were looking at putting the toxins on the apical side. So this is where they would be exposed in vivo. When the bacteria make them, they're going to be exposed to the apical side. And we saw that there was very slow toxicity when A or B was applied to the apical side of a nice, healthy, high barrier tissue construct. But when we looked and applied them just to the basal side, there was a very rapid drop. So the basal toxicity for A and B is much stronger than the apical side. Now, why, we don't know, but we do know, and there's some literature showing the localization of the receptors. Sometimes they're on actually in between the cells. Some are on the apical side, but very few. Some are unknown. But this could reflect different localization of the receptors, and that's one hypothesis why this happens. So maybe, clinically, you can imagine if you keep somebody or someone comes to the hospital with a nice, healthy barrier, they might be fine to more resistant to infection or the effects of these toxins than if somebody comes in with a leaky barrier for whatever reason. And again, when we looked at each of these tissue constructs from the apical side in the top row or the basal side in the bottom row, and look for cleaved caspase 3, which is an apoptotic marker, we saw very few dying cells in the apical compartment and quite a few on the basal side. So this also indicated that it was not just causing a leaky barrier, a higher leaky barrier from the basal exposure, but also killing the cells at a faster rate. So the take-home message and the thought from this is that with the increased expression of these TCDA and receptors and mature absorptive colonocytes and stem cells in the descending colon is different, and there's much more receptors in the differentiated cells. There's an uncoupled apical basal toxicity of TCDA and B, and that has some clinical implications, and differential toxin sensitivity between multiple healthy human donor intestines. So this again points out the idea. You have to have, if you're going to use these primary cultures, then you need to look at a lot of different people. The last two slides I have is we're trying to fix this. We're trying to develop new drugs to neutralize these peptides. These are my collaborators. 3 of them are from NC State. Carol Hall is a computational chemistry person. Stefano Menegatti is a peptide chemist. I am a gut models person, as I just showed you. And Nathan Crook is a probiotic delivery. He has these amazing ways to engineer new probiotics. And what Carol did is she computationally found a region of the catalytic domain of toxin A that she designed a short peptide to, very short, an 8-mer. Stefano made that 8-mer, and I put that on our cells with toxin A, with the idea that the peptide would bind that functional domain of the toxin, neutralize it, and it wouldn't have an effect. And when we did that, you can look here, the media alone, the negative is in black. When we added the TCDA alone, we had a significant drop in TIR in the yellow. And when we added the peptide, we nearly rescued the barrier function effects. So it neutralized the effect of those toxins. And I'm not going to show you the rest, but we have we've done this for TCDB now, and we have a whole panel of new peptides that bind these active sites of toxins that we hope can then be basically delivered through these probiotic methods that Nathan Crook is going to use. And then this is my funding and all the people in the lab that did the work. I'd be happy to answer any questions you might have.
Over there and take.
Sorry I ran over.
Yeah, no problem. Thank you, Dr. Magnus. So it's open for questions, do we have any questions or until, okay, Dr. Yen?
Okay, so thanks Scott for a very wonderful talk. Amazing. So very basic question. I probably missed something. So you said that there's something difference between the short chain fatty acid and long chain fatty acid when they absorbed by the colonocyte. So it seems to me that the short chain fatty acid can just directly pass through from the intestine lumen through those colonocytes and then go to the blood. It seems to be like it passes without any barriers.
Yes.
Long chain fatty has to be converted to short chain fatty acids through the mitochondria first, and then somehow then they have to be resynthesized into triglycerides, then packed into the chylomicron, and then goes to the blood for transport.
Right.
So if that's the correct sort of description of my understanding, why there's so much difference there? Why the long chain has to go through so much hectic?
Yeah, so the long chain fatty acids, when they get into the cell, they either go through beta oxidation to shorter chain fatty acids that if they go down to less than C8, they can basically just then go out. They don't have to go into chylomicrons at all. They go directly out the basal side and into the bloodstream. And the big difference is when the long chain fatty acids get into the cell, they can be reesterified, basically back in triglycerides. Then the triglyceride is packaged into a chylomicron, and the chylomicron goes into the lymph. And so it's a different way of handling these different chain fatty acids, likely because the longer the chain fatty acid, it has to have a mechanism to get out of the cell because it can't diffuse. It's too hydrophobic to just diffuse through the membrane and has to go through another mechanism of chylomicron formation and then chylomicron export. Whereas if it goes through beta oxidation to a certain chain length, then that fatty acid likely won't get put back into a triglyceride form and then will go directly into the bloodstream.
Now, in both scenarios, does there a fatty acid transport or receptor needed? For example, in the liver, we studied with CD36, for example, is required to transport the fatty acid into the hepatocytes. But I don't know, in the colonocyte, those short chain fatty acids, long chain fatty acids, they can just diffuse into the cells?
Yeah, so yes, they can diffuse in, but we don't know. Well, actually, we don't know if it's simple diffusion or if it's active transport or both. That's something that we really wanted to explore, still on the list of things to do. In fact, we were going to knock out some of the like CD36, some of the things that we thought could tell us if it's simple diffusion or if it's active, and we just haven't had time to do that. But those are some things we can do. We've developed, we published a paper a couple years ago on efficient CRISPR editing of these stem cells. And so it's a pretty easy thing for us to do now is these gain and loss of function experiments to answer questions just like you pointed out.
Thanks. Sean, you want to ask a question? Not sure.
Yeah.
Oh, yeah.
Thank you.
Sean?
Hi. Yeah. So
Hello.
great. Thank you for a very interesting talk. Enjoyed it. I have two quick questions. One is regarding the small peptide that you showed at the end.
Yeah.
Have you incubated the organoid or culture, 2D culture or 3D culture with just the peptide to see if that interacts with any of the receptors, perhaps mimicking the
Yeah.
sin?
Yeah, we're trying to figure out the mechanism of the neutralization of the toxin right now. The engineers have done some peptide plus toxin only studies to look at interactions and how long that the peptide will actually stay in the pocket.
Yeah.
And it's actually quite long. So in vitro in a tube, we know that it's binding in the right spot. And then in vivo, what we did is we incubated the peptide plus the toxin prior to putting it on the cells. We also pre-incubated the peptide just with the cells. So, which is just to hedge our bets to see where we could get the most neutralization effects. We don't know now how it actually works. So if we put the peptide on the cells prior to putting toxin, we don't know if it gets into the cell and then it keeps it, that's where it's interacting with the toxin domain inside of the cell, or is it happening on the outside of the cell? And this is a big area of interest, especially in vivo, because these peptides, if we were to give a probiotic that's expressing the peptide, it's going to be expressing that into the lumen, interacting first with the mucus layer, and then have to interact with the toxins at that level. So there's a lot of kind of like dynamics that we need to figure out as to how it's actually working at a pharmacological level.
I wonder if you could make it non-cell permeable version of that peptide to see if
that's exactly what we were going to do, is to put some sort of moiety on it to make it not get into the cell and see if that would help, or not work or work. And
so the other question I have is in relations to the single cell transcriptomics that you showed from different regions in the small intestine versus colon.
Yes.
And I was wondering if you see any differences in the expression signatures from, say, duodenum, jejunum, and ileum, even though they're all part of the small intestine. Are there some cell intrinsic differences depending on where they're coming from spatially within small and large intestine?
And are you asking that question from the perspective of fatty acid handling genes or anything?
I mean, in general, because
so in the manual, you can go to the paper. I didn't put this in the talk, but there's a section looking at each of those regions for subsets of genes that are involved in certain processes. Like we have looking at brush border enzymes, for instance, and how that those change along the proximal distal axis of those three regions of the small intestine. We look at we look at a lot of stuff. So some of the categories of bile acid transporters, we look at that, and you can see them come up in the ileum where they should, and they're less in the proximal part. So there are regional differences that you can see along that tract. And of course, we only took 3 regions. We didn't take a smaller like subset. It would have been really cool if we had the money to do that, to get a much higher resolution map. But there are clear transcriptomic differences that are regional to those. Even though it's one big long tube, there's definite differences in magnitude of gene expression along the tract.
So if I may so quickly ask you a follow-up, if you can sort of put these 3 different, say, small intestine derived crib cells in different order. Let's say you put the ileum before the duodenum in a continuous lineage layer in your manifold, right?
Yeah.
Using a bigger insert. Now, would they change plasticity, or do they remain their intrinsic?
I love that question. I don't know. That's a really cool question to ask, is if we made a long tube but reverse the order.
Continuous layer, right?
Would that work? Now, there's a lot of evidence coming out that cells in a certain regions have intrinsic sort of memory as to where they are from. So you have chromatin states in the proximal that are different than the distal, at least at the stem cell level. So and same in the colon, like if you look between colon and small intestine. So there's probably some, epigenetic memory that's there that won't allow them to totally convert, but you never know, right? The experiments haven't been done to really do the mixing and matching in a context that would have all the extrinsic influences that could change the chromatin states.
You have a unique tool to address that, actually.
Yeah. Yeah.
Interesting. Thank you.
Yeah.
Yeah. Do we have more questions? So then probably I can ask question,
Oh, yeah.
So Scott, your model is impressive, physiologically, I'm just, blown away. So my question is, if you want to go closer to physiological conditions, apical side of the intestinal cells, especially in small intestine, is exposed to fluid and movement all the time. So we have now like a chamber system. Lots of people who do endothelial biology use this chamber system with a movement on the apical side and measure what's happening on the level of the cells.
Yeah.
My question is like, perfecting your model, because that may influence like cell composition in different parts, and especially lipid absorption, like what is the speed of the movement.
Yeah.
And the other question, if you can comment, there are very specific diarrheal diseases related to dysfunctional lipid absorption. And one of them is related to mutations in DGAT1 gene, which is responsible for initiation and formation of lipid droplets. So can you model and affect children, which is highly significant for pediatric population?
Yeah.
Can you consider looking what is situation with the lipid droplets regulation in small intestine and eventually branching out to diseases like, in children related to lipid absorption?
Yeah. So the first question about basically having flow over the epithelium, that's definitely something we want to do. There's obviously shear forces that happen from not only just the fluid that's flowing, but it also involves peristalsis, right? So that's what's creating the force, is that, and then you have flow. So there's those two different forces. One's coming basically from under the cell, one's coming from on top of the cell, and those things have to be modeled in a way that we know are accurate. And that's the hard thing, right? How do you know what those forces are? And there's some literature looking at force of flow, peristalsis forces, things like that. But to get it into the right system, so there's a system called Emulate. I don't know if you're familiar with that, but it's a system that basically a device that contracts, that kind of makes this peristaltic motion, and they also have flow over the cells. So there is a device out there, but to my knowledge, they don't really have the capacity to culture primary cells. So now you have a system that can do those two things, but then you're growing a Keiko cell on it that may not be reflective. So there's tons of optimization, and we like to think about it from the perspective of what is the question we're trying to answer, and do we need all of those things? Sometimes we don't know, right? So we don't know, does peristalsis and flow, shear force flow, really change the fundamental property of what I'm trying to study? Maybe, maybe not, but we don't have it, and that's the caveat. We don't have it now. Once we have it, we'll put that in the system, probably redo all of the sequencing experiments I told you with just flow, with just peristalsis, and say, what are there fundamental transcriptomic changes that happen that will change how we interpret the model? And that's, again, any sort of ex vivo model, that's what you do. And these are very deconstructed systems. I mean, that's the caveat, right? We either don't do the experiment, or we do it in a somewhat contrived system with the knowledge we have to interpret it with the caveats. And so that's that. And then the other question was related to
diarrheal diseases related to lipid absorption.
So that's what's really beautiful about the system is we could, if you had a biopsy, from one of these children, we could actually culture the cells as stem cells and put it on there, or we could do a CRISPR-Cas9, mediated insertion or gene editing to mimic that, and then do the same thing on the system. And we've got that all down. So that's something that, if you want to collaborate,
That
we
would be
can
wonderful.
certainly do those sorts of things.
Yeah. Thank you, Scott. And if we don't have any questions, we can drop this grand rounds. Thank you, everybody, for attending and asking questions. And thank you, Scott, for coming in.
Thank you for having me.
Thank you very much.
Thank you.